Okay, this is the second seminar in Bioengineering Seminar Series 599J. I know some of the evening degree students are coming on campus to listen to the seminar in person rather than on video or on the internet. So make sure that you write your name down, and those of you who attend the seminar in person, all you have to do is write your name down rather than sending me a one-page abstract. Okay. So today's speaker is Professor Dan Beard, who is a research assistant professor of bioengineering since January of 2001. So it's been about 16 months since he joined the bioengineering faculty. Before that, for about 3.5 years, he was a Howard Hughes postdoctoral fellow at Courant Institute at New York University in Manhattan. And before that, he got his PhD in bioengineering in UW in 1997. And before that, in 1993, he got his BS degree in biomedical engineering from Boston University. So he finished his PhD in 4 years. Those our PhD students, that's a kind of model you should aim for. And he has been doing not only mass transport in physiology research, but as a Howard Hughes postdoctoral fellow, he has been doing a lot of molecular modeling. So he can cover both areas. And today he will give us a presentation with the title of In Silico Analysis of Integrated Cell Systems. David.
Thanks a lot, Dr. Cannon. So some of you may have noticed that I put in the title of my talk all the buzzwords and jargon that I could possibly fit in. And part of the reason for doing that is I want to, through the talk, take some stab at defining some of these terms, or you know, at least my best stab at defining them. So in silico is a term that just means with a computer, right, or computational. Cell systems is a recent buzzword that we hear a lot about. Systems biology was on the cover of Science magazine last month. On campus here we have in the bioengineering department the Cell Systems Initiative, and just down the road we have the Institute for Systems Biology. So we're very much in the systems biology era, whatever that is. So what is it? Well, one way of defining systems biology, not necessarily the best way, but one way is to take all of these words, and you could probably think of some more words that are related to biology and physiology. You probably all know what these words mean. So in systems biology, what we do is we add omics to the end of all the words. Okay? So if you used to be a cell biologist, and you used to do cell biology, well now you're doing cellomics and you're doing systems biology. Okay. Exactly. So anyhow, I didn't make any of these words up, but I made this one up. I think it might be out there. So this one's kind of tongue-in-cheek, but you heard it here first. Biolomics. Anyhow, to get a little bit more concrete in what we're talking about in systems biology, and I guess I changed the title here to cell systems modeling, what we're interested in is biological function. Okay, and there are a number of ways to get at biological function. One way is by looking at the anatomy, and anatomy is another one of these old-fashioned words. So we now do structural biology instead of anatomy. And this pathway to getting at biological function, I include in this pathway genomics, bioinformatics, you know, data mining. These sort of things I am categorizing as anatomy because genome sequence is really just a specialized part of cellular anatomy. So this is a very powerful pathway to getting at cell function because very often we can infer from structure biological function. Well, there's another route, and this is sort of the traditional bioengineering type pathway to understanding function, which is grounded in physiology, biophysics, biochemistry. What we do is we develop mechanistic models or mathematical models which describe how the biological system operates. Now, in the cell systems or systems biology era, what we need to do is we need to move through both of these pathways simultaneously. We need to build computational models and mechanistic models which integrate the large-scale data in some useful and practical way. Okay. If we can build models that are practical and useful at not only describing these data but predicting other data, then we can use large-scale integrated systems models to evaluate healthy and disease states, possibly identify and evaluate potential drug targets, and design optimal treatments. So this is one person's view of how we build integrated systems models of cell systems. And we start with looking at single cells, and this is where we're going to focus most of the talk today, is at modeling single cells, in particular a prokaryotic cell, and trying to characterize the biochemical behavior of intermediary metabolism of a whole cell system. And I'll introduce the constraint-based approach to modeling very large-scale systems. Now, the other thing we have to do is we need to develop, in order to develop integrated cell models, we need to characterize physiological systems. In particular, transport and microvascular networks is coupled in prokaryotic systems to metabolism and the biochemical behavior of cellular systems. And in the long run, what we want to be able to do is we want to integrate our cell systems modeling with our physiological systems modeling and build multi-scale integrated tissue systems, but that's a topic for a later talk. So like I said, today I'm going to focus on biochemical network analysis. The goals will be to predict the behavior of large-scale biochemical networks, in particular metabolism. If we can develop a theoretical basis for understanding how regulatory and control mechanisms operate in these systems, then we can understand what happens when you perturb part of the system, when you knock out a gene or if you block a pathway. How does the rest of the system respond? And with this kind of predictive power, we can hopefully identify drug targets and even design drugs. Now, there's a number of commercial ventures which are very interested in doing large-scale metabolic network modeling. And one of them is Integrated Genomics, and the company was a very successful company founded by Professor Evgeny Selkov, who is Argonne National Lab. And what Integrated Genomics provides is a very sophisticated genome annotation. And with that sophisticated annotation, they reconstruct the metabolic network of a prokaryote, or they reconstruct the metabolome of a prokaryotic cell. And then what do they do with it? They do a lot of things. They can hopefully predict the most probable control mechanisms. They can design organisms in order to produce some sort of product. And another interesting thing, they can list, they can identify promising targets for drugs in order to kill some sort of pathogenic strain. So I lifted this information off their website, integratedgenomics.com. Now, there's a lot of these companies, and at least another one that I know of is a recent startup in San Diego, Genomatica, which is founded by Bernard Paulson. And this is a blurb I listed from their website, and it's referenced right here. Bernard Paulson is the most visible leader in the field of bionetwork simulation and what he calls in silico biology. So I'm going to review a lot of what Bernard has done and contributed to this field, and you can read more about the commercial venture if you want to go to their website. So first, what is a biochemical network? A biochemical network is a collection of biochemical reactions. These reactions are interrelated. In general, the reactions can be compartmentalized. They might be distributed in space. Now, in developing tools for analyzing biochemical networks, it's important important to realize this interconnected web of chemical reactions is in principle, at least in some ways, similar to an electrical circuit network. So a lot of tools that have been developed for analyzing biochemical networks are developed in analogy with electrical circuit analysis. However, you have to realize that when reactions involve multiple species, the topology cannot be represented on a 2D graph. And so that means it's not straightforward. There's no straightforward way to apply the regular rules of electrical network analysis, Kirchhoff's loop laws and Kirchhoff's current laws, because they're based on looking at loops and nodes. Now, if you're going to develop an approach for studying a biochemical system, you have sort of two major choices. One is you can choose to characterize the detailed kinetics of the system, or you can adopt a constraint-based approach. And an example of a constraint-based approach is flux balance analysis. And I'll describe in detail what flux balance is in a minute. Now, the kinetic approach, that is, you write down a list of ordinary differential equations which describe how the concentrations of all the species evolve. It gives you very detailed predictions of all the fluxes and all the concentrations in the system. However, for very large, complex networks, and an entire organism scale network is very large and complex, the kinetic approach is going to require a tremendous number of parameters, and you don't know the parameters. And we'll see an example in a minute. Now, the constraint-based approach does not require you to use unknown parameters. It's based in writing down constraints which are based on hard physical laws such as mass conservation, energy conservation. However, these constraints are often not enough to precisely specify a solution. Therefore, if you can identify more constraints, then the smaller your feasible space becomes, the more accurate your solution becomes. So in general, the more constraints you have, and this thing's going on, the more constraints you have, the better off you are. That is, as long as you can identify constraints that are correct. So here's an example of a biochemical reaction. Okay, this is phosphofructokinase, which converts 6-carbon sugar with 1 phosphate to a 6-carbon sugar with 2 phosphates. If we wanted to characterize the flux through this reaction, we might write down an equation that looks something like this. Okay, and this is an approximation for the steady state flux through this reaction. It's very complicated. There's a number of unknown parameters, all of these KMs, the KM for the enzyme, there's a KM for magnesium binding to ATP, and magnesium binding to ADP, et cetera, et cetera. It's a very complicated system. Involves a number of free parameters that we just don't know. So where does that system, phosphofructokinase, fit into a large-scale metabolic system? Well, this is an example of a metabolic map. It's very large and it's very complicated. Right? So if we want, we could zoom in somewhere around here, and here's phosphofructokinase. Okay, it's in there somewhere. That's the point. Okay, but the point is that if we wanted to characterize on the level of kinetics the entire network, we'd be hopelessly lost. We have no chance of doing it, at least not anytime soon. So we move to a different approach, a constraint-based approach, which does not require that we write down these complicated expressions for every single reaction in the system. Okay, how does a constraint-based approach work? Well, I'm going to show you how it works for a very simple reaction system. This is a simple made-up example with 5 reactions, and there's 4 species, A, B, C, and D. The kinetic equations that you would write down for this system look like this. So we have an ODE system for concentrations of A, B, C, and D. And ddt of the concentration of A is equal to -1 times the flux through reaction 1, J1, plus 2 times the flux through reaction 2, because A comes in with a 2 stoichiometrically in this reaction, -1 here because A is on the left-hand side of reaction 3, et cetera, et cetera. And we can write down this system for any biochemical set of biochemical reactions. However, we don't know all these J's. Okay, we don't even know the functional form of all these J's. Never mind any unknown parameters that are associated with them. So what do? What we do is all I do here is I rewrite this equation as I put the concentrations in a vector, and then I put all of these stoichiometric coefficients in a matrix times the vector of fluxes. So this matrix is going to be called the stoichiometric matrix because it lists all of the stoichiometric coefficients. And in the steady state, we have this linear algebra statement, matrix S times the vector of fluxes J is equal to 0. Okay. Now for a typical system like metabolism, you might have 1,000 reactions and you might have several hundred metabolites. So what that means is you're going to have several thousand unknowns and several hundred equations. So there's not enough equations to specify all the unknowns. The system is underdetermined. So what we do is we treat the steady state statement as a constraint. Okay, the fluxes have to balance stoichiometrically. So just a list of some definitions that are involved in flux balance analysis. We have S, stoichiometric matrix, I already said, stores the stoichiometry of the reaction network. J is our vector of fluxes. S times J is equal to 0 is our statement of mass balance. It's our flux balance constraint. I'll get back to these inequality constraints in a second. So you've seen this before. You have a network of reactions. You can write down a stoichiometric matrix. In addition to the components of all the internal reactions, you have boundary fluxes here that we put in as columns like this. So this blue column is the flux associated with the input of A into the network. And for a prokaryotic system, you know the constraints on this flux because you can measure them in a bioreactor. You know how fast the E. coli cell can absorb glucose or can absorb oxygen, and you know based on what media it's growing in. So for the boundary constraints, you have a number of inequality constraints which you can apply based on sort of what type of system you're trying to model. So we have constraints. We have equality constraints, we have inequality constraints. If we want to simulate how a cell behaves, what we need is an objective function. Okay? We need to pick a flux from this feasible space. So this is work that Bernard Paulson did. Is what he and his students hypothesized was that the objective of the E. coli metabolic network is to produce biomass. And they know what all of the various components of biomass are, and they know the stoichiometry of all the various components because it can be measured experimentally. And then you know, say they're running a simulation with glucose media. They have glucose as a carbon source. They might have oxygen in the media, and they might have other small ions. And then we have the stoichiometry of all the growth factors that go into biomass, which are experimentally determined. And then in order to simulate fluxes, what we do is within the constraint space, we just pick a flux or a flux vector which maximizes the production of biomass. It's a pretty simple idea, but it's very powerful in practice because what Paulson has been able to do is show that flux balance analysis is about 90% accurate in identifying the essential genes of E. coli. Okay, how do they do that? Well, they go through and they knock out a gene, and then they ask the question, can this cell produce biomass? If the answer is no, then they say, well, that gene is essential. And 9 times out of 10, they're right. A number of times they're not right. And what we're seeing in this chart is a list of all the different genes, and then we're looking at prediction of cell growth on different media, glucose media, glycerol media, etc. A minus means that the cell cannot grow. The first minus refers to the experimental case, and then the plus means that the cell can grow in the in silico case. So these sort of red boxes are pointing out cases where essential genes are not identified. That is, the cell can't grow experimentally, but in the computer, when we knock the gene out, it can still grow. So sort of some of the theory that we've developed, we've been able to shed some light on these false negatives, okay, these red boxes here. So what theory have we developed? Well, flux balance analysis you can think of as being analogous to Kirchhoff's current law. It's just conservation of mass. And Kirchhoff's current law is a conservation of current. Okay. However, what is Kirchhoff's current law? Kirchhoff's current law looks like this. It says that if you have some flux into a node, flux J1 plus J2 has to be equal to this total flux here. Now, Kirchhoff's current law does not disallow solutions like this. Okay, as long as this flux, so in this case, this flux J1 minus J2 is equal to this flux J. Okay, so this system doesn't necessarily violate Kirchhoff's current law. Okay, what does it violate? Well, if these are passive resistors, it violates Kirchhoff's voltage law, and we have to be able to exclude solutions like this. So solutions like this are not excluded from, based on flux balance analysis. So what we've introduced is what we call energy balance analysis. And what is energy balance analysis? Well, let's look at a simple biochemical system again, 3 species and a simple cycle. Okay. Now there's a loop law for this system, and it says that the chemical potential, delta mu1, delta mu2, delta mu3, all sum to 0. Okay. So what does that mean? Well, that means that there can't be any flux through this system in steady state. Why is that? Because well, if there was a flux J going through this loop here, conservation of mass says that that flux could be anything you want it to be as long as it's equal in all of these arms of the loop. However, conservation of energy says that the chemical potentials all have to sum to 0. Now, if you were to, now the second law of thermodynamics tells you that the flux times delta mu has to be negative. So if you had so in order for there to be a non-zero flux going through this loop, all of these delta mu's would have to have the same sign. Okay, and the only way all of these delta mu's can have the same sign and sum to 0 is if they're all 0. And if they're all 0, well, that is the thermodynamic definition of equilibrium, which means there's no flux. Right? So that's circular logic for a circular system. Okay. So how do biological systems get flux through loops? Well, the way they do it is they couple one of the reactions to some other reaction. So you can think of this as a model for ATP going to ADP or whatever. And in this case, the loop law looks like this. Okay, we have a right-hand side. This is like a battery. We put a battery into our electrical circuit. Now, we can have a flux in this case. If we multiply this equation through by that flux J, what we get is a statement that says that the rate of heat dissipation that's dissipated by traveling through this cycle is equal to the rate of energy that's pumped in here. Okay. So this is energy balance for very simple loops that have only one species unimolecular reactions. Now, when you're looking at very complicated systems, and again, I'm going to go back to the same example here. This system has multi-species reactions, so there's no simple, straightforward way to identify the loops. Okay, again, we have the same stoichiometric matrix. So what we do in order to develop sort of a Kirchhoff's loop law for this system is we compute the null space vectors for this matrix. And the null space vectors are the vectors, are a basis of vectors where if you multiply S times these vectors, you'll get 0. Okay, well, what good is that? Well, if you take, say, one of these null space vectors and you multiply each reaction times all of the components of this null space vector and sum them, you'll get a perfectly balanced chemical equation. And the chemical potential for this equation has to be 0 by the definition of chemical potential. So to generalize that, what we do is we build a matrix K from a complete basis of the null space vectors, and that matrix K times the vector of chemical potentials has to be 0, and that's the Kirchhoff loop law. The second law of thermodynamics tells us that fluxes have to go down the chemical potential gradient, so J sub I times delta mu sub I is negative. Okay, so we need to consider these energy balance constraints in addition to the flux balance constraints in order to obtain fluxes that are thermodynamically feasible. And we have a paper in press. I can get you copies if you want. Okay, so what good does that do us? Well, with energy balance analysis, which I just introduced, the second law provides a nonlinear constraint that links the chemical potential and the flux. Okay, well, what good is that? Well, like I said earlier, the more constraints you have, the better. Combining energy balance analysis and flux balance analysis results in unique optimal solutions. It turns out this isn't always true. Sometimes it results in unique optimal solutions. But the fact is it results in a much smaller feasible space. And if you just apply flux balance analysis, you're 99.99% guaranteed to always predict thermodynamically impossible results. So you need energy balance analysis if you want physically realistic results. So here's a simple example. Same system again. We ask the question, what is the optimal rate at which this cell, imaginary cell, can produce D? What's the rate it can produce D? First of all, per mole input of A. Okay, well, flux balance analysis gives us a solution, and it says we can produce 1 mole of D for every mole of A input into the system. If we include the energy balance constraints, we get the same optimal production of D, but we get a very different flux vector. Okay, it turns out this flux balance solution is not thermodynamically feasible. And this, using energy balance analysis, we now have more information. If this was a real cell, we would know which pathways we might want to block and which pathways we might want to upregulate in order to get this cell to produce some product D. And we have similar results for other inputs. The other thing that energy balance analysis gives us is if we can predict chemical potential and chemical flux, we can predict conductance. And again, this is just an analogy from electrical circuits. Conductance is just the flux divided by the potential. We can, for a very simple reaction here, unimolecular reaction, we can derive a nonlinear relationship between the potential and the flux. For small fluxes, we can linearize that and get sort of an Ohm's law for biochemistry. And the Ohm's law for biochemistry says that the conductance changes as you change these rate constants. Okay, so a gene regulation or a change in enzyme phosphorylation or some sort of upregulation doesn't directly affect the potential, and it doesn't directly affect the flux, but what it affects is the conductance. And if we can estimate the conductance, then we can understand what's sort of the control mechanisms that are operating in a network. And that's precisely what we've done for the E. coli system, which was provided to us by Bernard Paulson. Here I'm listing some of the fluxes that are involved in the glycolytic series, and these are the predicted fluxes, potentials, and conductances for the wild-type simulation on glucose minimal media. And then what I've done is systematically knocked out pyruvate kinase, and these are the predicted fluxes. Pyruvate kinase is a non-essential gene, and you see when we knock out pyruvate kinase, none of these other fluxes are upregulated or downregulated. I'm coloring in upregulations green, downregulations as blue, and flux reversals as yellow. Okay, so this is the fluxes change, but the system responds passively. There is no sort of regulatory demands on the network. You knock out this gene, the cell doesn't care, it just goes on growing very happily. Phosphofructokinase is a very different example. If you knock out the phosphofructokinase, in order to grow, the cell requires almost all of the pathways either to be upregulated or downregulated or something to happen. So we hypothesized that these regulatory mechanisms just aren't available in E. coli, reason being that we know experimentally that phosphofructokinase is essential for growth on glucose media. So just to summarize, what do we learn from energy balance analysis? Well, one thing is we get physically realistic results. Even more interestingly, we get energy balance reveals insight into the regulatory and control mechanisms. So essential knockouts of essential genes place heavy demands on the metabolic regulatory mechanisms. When you knock out a non-essential gene, few or no regulatory steps are required, and it turns out that the network is robust. It's tolerant to these kinds of deletions. Now, one other thing that we can do with energy balance analysis is we can identify which reactions are reversible and which reactions are irreversible. This is the data structure which you can obtain from Bernard Paulson's website, and it's referenced down here. And you might not be able to read everything. It doesn't matter that much. Basically, what's important is some of the reactions are specified as being reversible, and those are colored in white, and some of them are specified as being irreversible, and those are colored in yellow. And this mostly comes from sort of a priori knowledge about how these biochemical systems operate. We know which of these pathways sort of tend to be irreversible, and which tend to be reversible. Of course, there's no such thing in biochemistry, there's no such thing in chemistry as an irreversible reaction. In principle, all reactions are reversible from a thermodynamic perspective. Now, what we can do with flux balance analysis is we can cut out all of these irreversibility constraints. We can allow all the reactions to be reversible. And that's what I've done in this experiment here. So let me explain what we're looking at. This is a predicted ranges of all the fluxes in the network, and I'm only looking at the first 30 or so, glycolytic series, pentose shunt, and TCA cycle. Okay, so plotted in blue is the predicted range of, might not be able to read this, but this is hexokinase, for example. And the predicted range for given growth and a given boundary flux, so a given uptake of glucose, a given uptake of oxygen, etc. According to the mass balance constraints, what is the range at which this flux can obtain? And it's completely unbounded based on flux balance analysis. And it turns out most of the fluxes based on mass balance or flux balance are completely unbounded, and we have no way of predicting specifically what they are. And that's what I've plotted in blue. Now in yellow, I've plotted the range predicted by energy balance analysis, and that allows us to narrow down the predicted behavior of the fluxes in the system tremendously, and it allows us to reproduce most of the inequality constraints. So that is, if it recovers information that we've thrown out of the system. If we don't know the reversibility or irreversibility of all the reactions in the system, well, under some specified production of biomass and some specified media, we can recover most of that information, which reactions are reversible. Now, if we put the reversibility constraints back into the system, I'm plotting the same thing here. In blue, the predicted ranges for flux balance analysis, and in yellow, the predicted ranges for energy balance analysis. And with these irreversibility constraints, the feasible space for flux balance analysis shrinks a lot, and only some of the reactions are unbounded. In particular, these phosphoglucose isomerase reactions are unbounded because we can just have arbitrary cycles which are don't affect sort of the net throughput through the network. But energy balance analysis further narrows down the predicted range and doesn't allow any of the reactions to be unbounded. So I want to mention that we have developed a prototype tool for simultaneously visualizing these networks and also simulating how they behave and predicting fluxes through the system. And although his name is too small here, Zheng Li is the architect of JSIM, and he's an architect of this tool for the simulation engine for modeling all manner of biological systems. And we have a prototype which we can show anybody who's interested later for doing biochemical network analysis. Now, before I finish, I'm going to try and finish up early, I just want to ask this question. Can our success at modeling these whole cell prokaryotic systems carry over to applications in human physiology? Of course, what we're interested in ultimately is not E. coli, or at least not what I'm primarily interested in. E. coli is a very convenient system. It's easy to manipulate. It's easy to compare our in silico results with experimental results. In